In this chapter, we'll be looking at the digestive system. Before we start, I wanted to do a review of this information that you've probably had during your chemistry courses, or biochemistry, or even during Anatomy and Physiology 1 or other biology classes. I wanted to review with you the main macromolecules that we eat and we need in our diet every day to be healthy, and then the components that they break down into. The top three here are carbohydrates. These are items that are big, huge molecules that your body can't make use of until you break them down into what are called monosaccharides or simple sugars. So an example of a carbohydrate might be starch, like from the vegetables that you eat, or carbohydrates in breads and grains. When you eat them, you break them down into their simpler sugars, which are monosaccharides, or examples of simple sugars include glucose, fructose, and galactose is another one. Simple sugars taste sweet, whereas in the carbohydrate form, they may not taste so sweet that way. Also, I'd like to remind you that another interchangeable word that we can use here is that another name for a carbohydrate or the type of molecule that it is, it's called a polymer. And a polymer is a big molecule that's made out of a chain of repeating units. So the repeating unit is the monosaccharide. So for instance, the carbohydrate starch, or another one is cellulose, and glycogen is another one. Those are basically long chains of glucose, but the molecules are different because of the way the glucoses are bonded together to make the chain. Proteins, those are big, huge molecules, very complex shaped 3D molecules. And I like to call them the workhorse of the body. They do so many functions, including what we've already talked about, how they can function as enzymes or as hemoglobin. They break down into the polypeptides, like for instance, hemoglobin has the 4 chains or the 4 polypeptide chains. And then those can break down further into their what are called the monomer units again, the amino acids. So again, proteins, polypeptides are chains of amino acids. Lipids and fats are not polymers. They're not made out of repeating units. They're not made out of monomers, but they are made out of fatty acids and a backbone molecule called glycerol. Fats, besides being delicious, of course, have, again, many functions in the body. We've already talked about a few, such as functioning as the steroid hormones like estrogen and testosterone and progesterone, also being the membrane of our cells, the phospholipid bilayer. So also very important molecules, also making up myelin of our nervous system. One I don't have mentioned here are nucleic acids, which make up our DNA and our RNA. DNA, RNA, big, huge molecules. They break down into what are called nucleotides. But these are the top three when we think diet and we think eating that we usually think of: carbs, proteins, and lipids or fats. Truthfully, in the form when they're in carbohydrates and protein and lipid or fat form, these molecules are too big for your body to do anything with them, too big to be able to build anything. So the function of your digestive system is going to be to break down these big macromolecules into their small monomer units, then send them eventually to the bloodstream, either directly to the blood, or in the case of what we learned already, that fats or lipids actually go to the lymph first and then go to the digestive system. But the job of the digestive system is to break those down, get them into the blood, send them to the cells, and then your cells will rebuild. For instance, those amino acids will put those amino acids together into the proteins that build you, or put together the glucose units or the monosaccharide units to build something that's needed within a cell. Like, for instance, perhaps the carbohydrate group on the glycolipids that make up our blood types. So the digestive system acts like a disassembly line, pulling apart these big, huge molecules to get them to the blood, then to the cells, to get them to be used in their useful form. The study of the digestive tract and the diagnosis and treatment of disorders of the digestive tract is called gastroenterology. In this first section, we've got a lot of objectives here. We should be able to list the functions and the major physiological processes of the digestive system, distinguish between mechanical and chemical digestion, describe the basic chemical process underlying all chemical digestion, and name the major substrates and products of this process, list the regions of the digestive tract and the accessory organs of the digestive tract, identify the layers of the digestive tract and describe its relationship to the peritoneum, and describe the general neural and chemical controls over the digestive functions. The digestive system is an organ system, quite a few organs to be exact, that processes food, extracts nutrients, and then eliminates residue. We wouldn't want any of that residue clogging up our machinery, so we do have to get rid of that. We have what's called a two-opening digestive tract, but some organisms have only a one-opening digestive tract. I bet you can imagine what they do to get rid of their wastes. But anyway, this is more about the human digestive system. There's 5 stages of digestion. Ingestion is the selective intake of food, and of course you have voluntary control over that and what you select to eat. Digestion is the mechanical and the chemical breakdown of food into a form usable by the body, like I talked at the introduction to this lecture. Absorption is the uptake of the nutrient molecules into the epithelial cells of the digestive tract, and then into the blood and to the lymph. Compaction is the absorbing water and consolidating the indigestible residue into feces, and then defecation is the elimination of those feces. Mechanical digestion is the physical breakdown of food into smaller particles, cutting and grinding with your teeth, the churning action of the stomach and the intestines all participate in mechanical digestion. And basically, it's to physically break those particles of food, which are big particles, into small enough particles that really expose their surface area so the digestive enzymes can get in there and do their job to break those big polymers or macromolecules down into their individual monomers. Chemical digestion is a series of hydrolysis reactions. A hydrolysis reaction is to break apart the actual monomer. It's carried out by digestive enzymes, which are produced by a number of different parts of the digestive system, such as your salivary glands create digestive enzymes for you, the stomach and the pancreas, and even the small intestine. And sometimes it's a number of different types of cells within these areas as well. So the results, again, carbohydrates, big macromolecules, will break those, another word for those carbohydrates, polysaccharides, into their monosaccharides. Proteins broken down into amino acids, which is its building block, and fats into monoglycerides and fatty acids. and then also nucleic acids like DNA and RNA into their individual building blocks called nucleotides. Some nutrients are present in usable form in ingested food and can actually be directly absorbed right away, like vitamins and minerals, amino acids, which are already broken down, cholesterol, and water. Our digestive system has two subdivisions, the digestive tract and the accessory organs. The digestive tract is also called the alimentary canal, and if you want to think of it this way, you can think of it as a big tube that goes through your body all the way from mouth to anus. If you wanted to, if you could think about it this way, you could actually thread a wire all the way down your mouth, down the digestive tract, and then out the anus. And it would be a complete trip all the way through. Although there's a lot of coils and a lot of turns in our digestive tract, but if you went to like a earthworm, it has a very simple digestive tract where its intestine is just a straight organ, where ours is very coiled. We have about 30 feet of digestive tract. In order to get 30 feet of stuff inside of you, it again has to be coiled and wound up nice and tight so that it fits inside your body. In order of the organs, if we were traveling down that tube, we would go past the mouth, the pharynx, the esophagus, the stomach, the small intestine, and then finally the large intestine till we reach the anus. The gastrointestinal tract, or the GI tract, is just the stomach and the intestine part. Accessory organs are organs that are kind of on the sides of the digestive tract, and they assist in digestion. Our digestive tract can't do it alone, so we need a little help from accessory organs like our teeth, tongue, salivary glands, liver, gallbladder, and pancreas. The digestive tract, if you want to think of it this way as well, is actually open to the environment at both ends. Most material in it has not entered the body tissues. It's considered to be external to the body until it's actually absorbed into the epithelial cells of the alimentary canal. So on a strict sense, defecated food that you couldn't digest, that was never considered inside the body. Strictly speaking here, of course. So if we go down our digestive tract and we look at the layers of the different organs, we're going to see about the same thing in each of those organs. They're all going to have a basic structural plan with about 4 layers. Some of them modified a little bit in different organs, but this is kind of the general plan. So if you learn this one, it'll help fit pretty much all of the organs of the digestive system. So the innermost layer is the mucosa right here. It's made out of the epithelium and then the lamina propria, which is the connective tissue, and then the muscularis mucosae. Then there's the submucosa, that is right here. Then we have our muscular layer, which consists of a layer of both circular and longitudinal muscles. That's here. The inner layer of muscle is circular, the outer layer is longitudinal. That's going to be important for propelling our food forward and getting it ground up as well. And then the serosa is the outermost layer here, kind of shown peeled away, consisting of areolar tissue and mesothelium. In this diagram, we can actually see them labeled as well as some other parts. Again, the lumen is the empty area in the middle. The mucosa, or the mucous membrane, lines the lumen and consists of a inner epithelium. Again, the lamina propria, which is the connective tissue layer, a muscularis mucosa, which is a thin layer of smooth muscle. When that tenses, it creates grooves and ridges that enhance surface area and contact with food and will therefore improve digestion and nutrient absorption. And this layer also has a mucosa-associated lymphatic tissue, also known as MALT, cleverly named there. This exhibits an abundance of lymphocytes and lymphatic nodules. The submucosa, moving our way outward, that is the thicker layer of loose connective tissue, has blood vessels and lymph vessels and other mucus secreting glands. We're going to find that mucus is very important to this system of the body, not only for lubrication but also for protection. Also has some malt in this area. The muscularis externa, again, has the two muscle layers. The inner is the circular layer. The circular layer muscles in this layer is important for forming what are called sphincters. Sphincters are tight circular muscles that can relax, or dilate, or constrict and tighten and close. And that helps to regulate the passage of material through the tract. We wouldn't want material going through if it's not ready, or we wouldn't want too much going through at once. The outer longitudinal layer is for motility, which propels food forward. The last outermost layer is the serosa. It's got the thin areolar tissue with some simple squamous mesothelium. It begins in the lower 3 to 4 centimeters of the esophagus and ends just before the rectum. The adventitia can be found here as well, which is the fibrous connective tissue layer that binds and blends the pharynx, most of the esophagus, and the rectum into adjacent connective tissue of other organs around it. The enteric nervous system is the nervous network in the esophagus, stomach, and intestines that regulates the digestive tract motility and secretion of different enzymes and substances and blood flow. It's said to consist of 100 million neurons, and it can actually function independently of the central nervous system, although your central nervous system does exert some influence on it, and often it's considered part of the autonomic nervous system as well, because much of what you do here you do not have to think about, thank goodness. The enteric nervous system is composed of two networks of neurons, the submucosal or Meissner plexus. This is in the submucosa. It controls glandular secretions of the mucosa and controls movements of the muscularis mucosae. Then there's also the myenteric or Auerbach plexus. Here the parasympathetic ganglia and nerve fibers are between the two layers of the muscularis externa, and it helps to control peristalsis, which is a contractile movement or a muscular pushing to propel items through the digestive tract. There's also the mesenteries. Mesentery is connective tissue sheets that help to suspend the organs like the stomach and the intestines from the abdominal wall. And they're kind of loosely suspended, which gives some freedom for all those contractions that your digestive system is going to perform. But it also helps to hold that abdominal viscera in the proper relationship to each other. We don't want our organs to get twisted and tangled up. It prevents, like I said, the intestines from becoming twisted and tangled, because think about how your body moves, how you, even just laying down, how you may move from side to side, on your back, on your stomach. This is going to help in getting twisted up. It also has blood vessels and often lymph nodes and lymph vessels as well. The parietal peritoneum is a serous membrane that lines the wall of the abdominal cavity. It turns inward along the posterior midline, which then forms the dorsal mesentery. That's a translucent two-layered membrane extending to the digestive tract. The two layers of that mesentery separate and then pass around opposite sides of the organ, forming what's called the serosa. They come together on the far side of the organ and continue as another sheet of tissue called the anterior or the ventral mesentery. This may hang freely in the abdominal cavity and might attach to the anterior abdominal wall or other organs. And when you look here, this is what they're talking about when they say mesentery. It's the reason why if you have lab and you dissected a fetal pig or some other organism and you inspected the intestines, you just can't grab them and unwind them and pull them out. You actually have to cut all this mesentery first to get them loosened up in order to unravel all of this. And here you can nicely see the blood vessels within there as well. In our next slide, we'll also talk about something called the mesocolon, which anchors the colon to the body wall, and that can be found up here. And then we go from this diagram, put those intestines back in, and we have this diagram. This is all overlaying the intestines. We have the lesser omentum and the greater omentum. The lesser omentum is a ventral mesentery that extends from the lesser curvature of the stomach to the liver. So that's right here. Here's the liver up here. Here's the lesser curvature of the stomach. And here's the lesser omentum here. Then there's the greater omentum. It hangs from the greater curvature of the stomach. That's here. And then covers the small intestine like an apron. Its inner superior margin forms serous membranes around the spleen and the transverse colon, which then that is called the mesocolon, which we learned back here, anchors the colon to the body wall. So like I said, you just can't pull these organs out. They're anchored in there pretty well, but yet loose enough so that they can have some freedom and flexibility to move when they're passing food and other materials through. These are words to describe some of the body structures and organs. If an organ is intraperitoneal, it's enclosed by mesentery on both sides, and it's considered within the peritoneal cavity. For instance, the stomach, liver, and parts of the large and small intestine are considered intraperitoneal. If it's considered retroperitoneal, the organ would lie against the posterior body wall, and it's covered by peritoneum on its anterior side only. So this is actually considered to be outside the peritoneal cavity. The duodenum, the pancreas, and parts of the large intestine, and even the kidneys, which aren't mentioned here, are retroperitoneal. So if you went in ventrally and went all the way to your back body wall, you would see your kidneys back there, but you would actually have to dig for them because they would be under this tissue. So regulation of the digestive tract, motility and secretion of the digestive tract are controlled by neural, hormonal, and paracrine mechanisms. For neural control, there's short reflexes. Usually stretch or chemical stimulation would act through the myenteric plexus. It stimulates the peristaltic contractions of swallowing. Then there's long reflexes, or vagovagal. These are parasympathetic stimulation of digestion, motility, and secretion. Hormones also help control this. Remember, hormones are chemical messengers created into the bloodstream that will affect cells or other target organs, usually or sometimes far away from the organ that secreted the substance or the gland that secreted the substance. Gastrin and secretin are two examples. Paracrine secretions also have an influence. These are chemical messengers that diffuse through the tissue fluids to stimulate nearby target cells. In this section, our expected learning outcomes or objectives are to describe the gross anatomy of the digestive tract from the mouth through the esophagus, to describe the composition and functions of saliva, and to describe the neural control of salivation and swallowing. So starting at the mouth, it's also known as the oral or buccal cavity. Its jobs are to take in food, that's called ingestion, the sense of taste and other sensory responses to food, chewing and chemical digestion. So we're going to start the mechanical and the chemical breakdown of the foods we eat here. Swallowing, speech, and respiration are also some functions. The mouth is enclosed by the cheeks, the lips, the palate, and the tongue. The oral fissure is the anterior opening between the lips. The fauces is the posterior opening to the throat. And stratified squamous epithelium lines our mouths. In some areas where food will contact a lot, we need a little extra protection, so we have the keratinized epithelium there. That's the epithelium that has the dead layer on top, little extra protein keratin to protect it. And in some other areas where we may not need as much protection from friction is the non-keratinized sort. Here you can locate some of the different features of the oral cavity. The cheeks and the lips have a job as well to retain food, push it between the teeth. We also use it for speech and sucking and blowing and suckling by infants. Its fleshiness is due to the subcutaneous fat, and there's a buccinator muscle in each cheek and an orbicularis oris of the lips. The labial frenulum is the median fold that attaches each lip to the gum between the anterior incisors. That little flap of tissue just right underneath your lips there. And then the vestibule is the space between the cheek or lips and the teeth. Your lips are divided into three areas. There's a cutaneous area, which is colored like the rest of the face, has hair follicles and sebaceous glands. There's the red area or vermilion area. It's a hairless region where the lips meet. Here there's tall dermal papilla that has blood vessels and nerves that come closer to the epidermal surface and give us that great sensitivity that we have in our lips. And then the labial mucosa is the inner surface of the lips facing the actual gums and teeth inside the mouth. The tongue is a muscular organ. It's pretty bulky, but still remarkably agile and sensitive. You can manipulate food, but even try to pick food out of your teeth with it. You can sense taste and textures. The lingual papillae are bumps and projections that are the sites of most of the taste buds. The body of the tongue is the anterior 2/3 of it and occupies the oral cavity, whereas the root is the posterior 1/3 of the tongue that occupies the oropharynx, which we learned about in our last chapter. And then there's also lingual glands, and these secrete a part of our saliva. The palate separates the oral cavity from the nasal cavity, like we learned last chapter, so that we can breathe while chewing food. Separates the nasal and the oral cavities. There's the hard or bony palate. It's the anterior portion that's supported by the palatine processes of the maxillae and the palatine bones. Then there's palatine rugae, which are the transverse ridges. If you just feel your tongue up on your hard palate, you'll feel those ridges up there. That helps you hold and manipulate food. The soft palate, if you're good, you can take your tongue and actually get it back there and feel your soft palate as well. It's a little more soft, spongy texture. It's made out of skeletal muscle and glandular tissue, but no bone. And at the back of it, there is the uvula, which is that little medial projection back there visible at the rear of the mouth. That actually helps retain food in the mouth until you're ready to swallow. During the swallowing reflex, food kind of sits way at the back of the tongue there, ready to be swallowed. The teeth, also known as dentition, masticate or chew food into smaller pieces. The idea is to make the food into a bigger surface area so those enzymes can get in there to do their job for chemical digestion. In an adult, there should be normally 32 teeth, 16 in the mandible and 16 in the maxilla. So from midline to the rear of each jaw, there should be 2 incisors. Those are chisel-like cutting teeth that you use to bite off a piece of food. 1 canine, a little bit pointed and acts to puncture and shred food. In some mammals, that canine is really pointed. There's 2 premolars. They have a broad surface for crushing, shredding, and grinding. And then 3 molars, which have a little bit more even, broader surface, still for crushing and shredding and grinding as well. The alveolus is the tooth socket in the bone, and the gomphosis joint is formed between the tooth and the bone. The periodontal ligament is a modified periosteum whose collagen fibers penetrate into the bone on one side and then into the tooth on the other. It anchors that tooth firmly, but yet allows it some flexibility for a little bit of movement for all that pressure with chewing. The gingiva is the gum and covers that alveolar bone. On the regions of a tooth, there's the crown, which is that portion above the gum. The root is just below the gum and embedded into that alveolar bone. The neck is the point where the crown, root, and gum meet. And the gingival sulcus is the space between the tooth and the gum. Hygiene in that sulcus is very important to dental health because bacteria from get food particles in there, bacteria could start inhabiting those areas, and the compounds that the bacteria make can actually degrade your bone or degrade your enamel on your teeth. Dentin is the hard yellowish tissue that makes up most of the teeth that's here. You can regenerate that, but the enamel up here covers it. You can't regenerate. The root canal is the space in the root leading to the pulp cavity in the crown. It's right here. Here, then you'll reach nerves and blood vessels as well. This picture amazes me of a child's skull. How they have their baby teeth here and their adult teeth ready to come in. Isn't that amazing? How those just kind of wait in the wings here of the skull. So the milk teeth or the baby teeth are actually called deciduous teeth. There's 20 of those. They develop beneath the gums and erupt in a predictable order. Any of you who have babies and children know this. From about 6 to 30 months it starts, beginning with the incisors, and between ages 6 to 25 they're replaced by 32 permanent teeth. The third molars, the wisdom teeth, erupt between usually ages 17 to 25. If there's not enough room for them because of crowding, We call them impacted, and then many people have to have them removed. The human mouth is home to more than 700 species of microorganisms, especially bacteria. If you leave sticky residue from your meals on your teeth, it can result in plaque. Calculus is calcified plaque. When bacteria metabolize the sugars that you eat and release acids, it can dissolve the minerals of your enamel and then form cavities. Root canal therapy is necessary if that cavity reaches the pulp. And gingivitis is inflammation of the gums from improper care of the teeth. And periodontal disease is the destruction of the supporting bone around the teeth, which can also result in tooth loss. Mastication is chewing, and it breaks food into smaller pieces to be swallowed, and then exposes the food to more surface area so that you can get in there and get those digestive enzymes working. The first step in food stimulates oral receptors that trigger an involuntary chewing reflex. Then we have saliva, which is another helpful thing in our mouths. It moistens the mouth. It moistens the food that we eat, binds it together in what's called the bolus, which helps us swallow it. It also contains some digestive enzymes to help start breaking down food so that we can actually taste it. For instance, when carbohydrates start to break down in our mouths, we taste the sugar, which actually tastes sweeter. Fructose is a monosaccharide. It's known as the sweetest of all the monosaccharides. Our saliva also inhibits bacterial growth and can help to cleanse our teeth. It's a hypotonic solution, about up to 99.5% water, and some of these solutes, like salivary amylase, it's an enzyme that begins starch digestion in the mouth. Lingual lipase, which we already talked about. This is an enzyme that is activated by stomach acid and digests fat after the food is swallowed. Mucus, which binds and lubricates a mass of food and aids in swallowing. Lysozyme, that's an enzyme that helps to kill bacteria. There may also be immunoglobulin A. That's an antibody that inhibits bacterial growth. And other multiple electrolytes like sodium, potassium, chlorine, phosphate, and bicarbonate. The pH should be between 6.8 to 7 in your mouth. The intrinsic salivary glands are small glands dispersed amid the other oral tissues like the lingual glands in the tongue, the labial glands inside the lips, the palatine glands on the roof of the mouth, the buccal glands inside the cheek, all of them secreting saliva at a fairly constant rate, but probably not as much as these extrinsic salivary glands. These are three pairs connected to the oral cavity by ducts. The parotid gland is anterior to the earlobe. On a little side note, if you get mumps, it's an inflammation of the parotid gland caused by a virus. The submandibular gland is located halfway along the body of the mandible, and its duct empties at the side of the lingual frenulum near the lower central incisors. And the sublingual gland is located on the floor of the mouth, and it empties posterior to the papilla of the submandibular duct. These secrete about 1 to 1.5 liters of saliva a day. Just think if you could collect all of that in a 1-liter beaker in one day. That is a lot. Here we can see some of those structures, like the parotid gland here, the submandibular gland here, and the sublingual gland there. For their histology, they have branched ducts ending in acini. These are secretory cells. The mucus cells secrete mucus. The serous cells secrete thin fluid rich in enzymes and electrolytes. And if it's a mixed acinus, then they have both secretions. Looks very tree-like. The cells of the acini filter water and electrolytes from the blood and add amylase, mucin, and lysozyme to our saliva. The salivary nuclei in the medulla oblongata and the pons respond to signals generated by the presence of food. They can be excited by tactile, pressure, and taste receptors. Salivatory nuclei receive input from the higher brain centers as well, such as walking into someone's house and having that great scent of food can get you salivating. Seeing the food, or even sometimes you just consciously thinking about food, will stimulate salivation. The mass of that you swallow as a result of saliva binding the food particles into that soft slippery mass is called a bolus. The pharynx is the muscular funnel connecting the oral cavity to the esophagus, and also the nasal cavity to the larynx, as we learned in our last chapter. It's where the digestive and respiratory systems intersect. It has a deep layer of longitudinal skeletal muscle and a superficial layer of circular skeletal muscles that form the pharyngeal constrictors. And it'll help force food downward during swallowing. Next, going down the digestive tract, we have the esophagus, which is a straight muscular tube about 25 to 30 centimeters long. It begins at the level between the C6 and the cricoid cartilage. It extends from the pharynx to the cardiac orifice of the stomach, and then passes through the esophageal hiatus and the diaphragm. The lower esophageal sphincter is often called the cardiac sphincter as well, and food pauses here for a brief moment because that sphincter muscle is constricted and closed and has to open and relax for food to pass through. That sphincter is very important. It keeps food from coming back up, and food that would be in the stomach would also have a acidic content to it, which could then harm or damage the esophagus lining, as the esophagus is not meant to hold acidic contents. If your cardiac sphincter is not working well, then you may suffer from heartburn or acid reflux. The esophagus has non-keratinized, stratified squamous epithelium, and it meets the stomach at the level of the T7 vertebrae. And it's also covered with the adventitia. Next, we'll take a look at swallowing. There's the oral phase, the pharyngeal phase, and the esophageal phase. In the oral phase, the tongue forms the food bolus and pushes it into the laryngopharynx. In the pharyngeal phase, the palate, tongue, vocal cords, and epiglottis block the oral and nasal cavities and the airway while the pharyngeal constrictors push that bolus into the esophagus. And then number 3, there's the esophageal phase, where peristalsis, those are muscular contractions, will drive that bolus downward. And then the relaxation of the lower esophageal or cardiac sphincter, which will then admit it into the stomach. Swallowing is also called deglutition. It involves about 22 muscles in the mouth, pharynx, and the esophagus. The swallowing center is a pair of nuclei in the medulla oblongata, and that coordinates the act of swallowing. It communicates with the muscles of the pharynx and the esophagus by ways of the trigeminal, the facial, the glossopharyngeal, and the hypoglossal nerves. Again, the swallowing occurs in those three phases, which I've also already talked about: the oral phase, the pharyngeal phase. Again, the oral phase, you can actually control the beginning of that and decide when to swallow. But once we get to the pharyngeal phase, that's involuntary. And then the esophageal phase, which is helped by gravity as well. Liquid can reach the stomach in about 1 to 2 seconds, whereas a food bolus would take 4 to 8. The interesting thing is that the peristaltic muscle contractions, as well as gravity, propel that food to the stomach. But if you were to stand on your head, the peristalsis would still ensure that the food would end up in the stomach. So yes, you can swallow upside down. In this section, we'll take a look at the stomach. Our objectives are to be able to describe the gross and microscopic anatomy of the stomach, to state the function of each type of epithelial cell in the gastric mucosa, to identify the secretions of the stomach and state their functions, to explain how the stomach produces hydrochloric acid and pepsin, to describe the contractile responses of the stomach to food, and to name the 3 phases of gastric function. The stomach is the muscular sac in the upper left abdominal cavity, just immediately inferior to the diaphragm. Its primary function is a food storage organ, but also helps with the mechanical mixing and the protein digesting. When it's empty, it's about 50 milliliters, but can stretch to 1 to 1.5 liters after a typical meal, and all the way up to 4 liters after that big Thanksgiving meal that you may often have, and sometimes extend nearly as far as the pelvis. That has to be uncomfortable. Mechanically, the stomach breaks up food and liquefies it. That helps that chemical digestion again by increasing the surface area. When you get that soupy mixture of semi-digested food in the stomach, it's called chyme. CHYME. Most of the digestion, though, is going to occur after the chyme passes to the small intestine. The stomach is J-shaped. It's divided into 4 regions. Here's the cardiac region, cardia, the fundic region, or the fundus, the body, which is the main portion here, and then the pyloric region in the end. We can also follow the pyloric canal here, the pylorus, also including the pyloric sphincter, which helps hold food in till it's ready to be passed to the small intestine, which is here. The first part of the small intestine is called the duodenum. Note the bulge of the fundus and the narrowing of the pyloric region and the thickness of the pyloric sphincter, and the greater, this is the greater, and then this is the lesser curvature. We can see how the greater omentum hangs from the greater curvature and the lesser omentum from the lesser curvature. Inside are longitudinal wrinkles called rugae. The stomach receives parasympathetic fibers from the vagus nerve and sympathetic fibers from the celiac ganglia. It's supplied with blood from the celiac trunk. And when blood is drained from the stomach and intestines, it enters the hepatic portal circulation and then is filtered through the liver before returning to the heart. The stomach has simple columnar epithelium. The apical regions of its surface cells are filled with mucin. Those swell with water and then become mucus after it's secreted. The mucosa and submucosa are flat when the stomach is full. but then form those longitudinal wrinkles called rugae when they're empty. The muscularis externa here has 3 layers instead of 2. We can see the 3 layers here. There's a longitudinal, a circular, and the obliques. The obliques kind of run at more of a 45-degree angle. All of these muscle layers here are important in how the stomach can churn. Gastric pits are depressions in the gastric mucosa and look like this diagram over off to the right. They're lined with simple columnar epithelium and have 2 or 3 tubular glands open into the bottom of each gastric pit. Some are cardiac glands, some are pyloric, and some are gastric. This is a remarkable picture of those gastric pits. Mucus cells, those secrete mucus. Mucus in the stomach is important for protection from the acidic environment. The regenerative stem cells found in the base of the pit and in the neck of the gland divide continually and rapidly to replace cells that die because stomach cells don't last very long. And the parietal cells, found mostly in the upper half of the gland here, are in charge of secreting hydrochloric acid, intrinsic factor, and a hunger hormone called ghrelin. Ghrelin is secreted by these parietal cells in the gastric fundus, especially when the stomach is empty. It produces that sensation of hunger and stimulates the hypothalamus to secrete growth hormone releasing hormone, which primes the body to take advantage of the nutrients that is about to be absorbed into the blood. And then within an hour after eating, ghrelin secretion ceases. There's also the chief cells here. They're the most numerous and secrete gastric lipase and pepsinogen. They dominate the lower half of these gastric glands, and they are absent from the pyloric and cardiac glands. There's the enteroendocrine glands, which are concentrated in the lower end of the gland. They secrete hormones and paracrine messengers that help to regulate digestion. You make about 2 to 3 liters of gastric juice a day, and it's mainly a mixture of water, hydrochloric acid, and pepsin. So let's talk about those molecules. Hydrochloric acid is also known as HCl. Gastric juice has a high concentration of this, which makes the pH very low or acidic, sometimes as low as 0.8, which is very low. You kind of already know how these cells, the parietal cell here, is going to make hydrochloric acid. If we remember this equation right there, we saw that in our last chapter. And here's the hydrogen we're going to use in the hydrochloric acid. So those parietal cells have an enzyme called carbonic anhydrase, which helps in this equation right here. The hydrogen is then pumped into the gastric gland by the hydrogen potassium pump. Then an antiporter, which is going to do kind of a little switcheroo, uses ATP to pump hydrogen out and potassium in. And then bicarbonate is exchanged for chlorine. That's again called the chloride shift. That's from the blood plasma. So then the chloride ion is pumped into the lumen of the gastric gland to join the hydrogen, and then it forms HCl, or hydrochloric acid. The elevated bicarbonate ion amount in the blood causes what's called the alkaline tide, which is a high pH of blood leaving the stomach. It makes the blood in that area there more alkaline or more basic, meaning a pH above 7. This happens when digestion is occurring and these cells are stimulated to be taking hydrogen from this equation. Hydrochloric acid is important for a number of reasons. It denatures the protein, so basically it takes the protein, which is a big kind of folded up molecule, and it unfolds it, which then allows the enzymes to get in and get at it. It also activates pepsin and lingual lipase. I'll jump ahead to this diagram here. Many of our enzymes have what's called a precursor molecule. So you can see the enzyme here, which our enzyme is going to be pepsin, but it's got this other molecule chain on it. So in this form, it's inactive, and all we have to do is remove this orange part, and then we'll have our enzyme pepsin. But we may not want pepsin to be located in that cell because it's going to start digesting proteins in that cell. So we want it in its inactive form. We'll pump that out of the cell. Hydrochloric acid will cleave off that molecule in orange, and then we'll have our active enzyme called pepsin, which then will start to break down the dietary proteins, not all the way into amino acids, but usually breaking apart the proteins into smaller chains, which then this will be finished later in the small intestine. Hydrochloric acid also breaks up connective tissue and plant cell walls and helps to liquefy food to form chyme. It also converts the ferric ions to ferrous ions, and you use the Fe2+ to make hemoglobin, and can actually destroy pathogens that end up in our stomach. So we could say that they're part of that non-specific defense in immunity. As I've already mentioned with pepsin, it comes as a precursor form first. The precursor form is called a zymogen. And if I ask why here, why start as a precursor again, we don't want those enzymes working within the cell because they'll start destroying our proteins and other molecules within there. We want to make them, but in a safe form, and pump them out of the cell, then put them into their form that's useful. There's other stomach secretions, including gastric lipase, made by the chief cells, and this digests only about 10 to 15% of the dietary fats in the stomach. The rest will be completed by the small intestine. We also make intrinsic factor, which is a glycoprotein secreted by the parietal cells, and this helps us to absorb vitamin B12. This is a very important vitamin that we use to synthesize hemoglobin. So if you're deficient in it, it causes anemia. Secretion of this intrinsic factor is an indispensable function of the stomach. You could literally remove the stomach and still get digestion done, but you would certainly miss the synthesizing of that vitamin B12. So you would have to take supplements instead. Gastric motility, that means the movement of the digestive tract. The swallowing center of the medulla oblongata signals the stomach to relax. The vagus nerve relays the message from the medulla and activates a receptive relaxation response in the stomach, which will make it relax to hold more food to stretch. Soon the stomach will show rhythmic peristaltic contractions, which the stomach has its own little pacemaker cells in the longitudinal layer of the muscularis externa. All we're going to need is some, just like in the heart, some sodium leaks. Then we'll have a little pacemaker. We'll also have a ring of constriction every 20 seconds, becomes stronger in the pyloric region. After 30 minutes or so, they'll become quite strong, and they'll churn and mix the food with the gastric juice to increase its surface area and start promoting chemical digestion. As we're digesting the chyme, only about 3 milliliters is squirted into the duodenum at a time. And this gives our small intestine time to neutralize that stomach acid that had hydrochloric acid in it. So the small intestine is going to be more of a basic or an alkaline environment. And it'll help digest nutrients little by little and make it more efficient that way. If the duodenum becomes overfilled, then it'll inhibit gastric motility. In a typical meal, it should be emptied from the stomach in about 4 to 6 hours, depending on what you ate. And if it was a liquid meal, it's going to obviously move faster. Now, if you've ever vomited before, you felt the effects of the peristaltic contractions, but in this case, they're moving in reverse. Vomiting is a forceful ejection of the stomach and intestinal contents from the mouth. You've probably also felt that burning sensation in your throat afterward, and that's that hydrochloric acid that came up with the chyme. The emetic center of the medulla oblongata integrates multiple muscle actions in order to make this happen. Vomiting is induced by many different things, including anything from trauma to overstretching the stomach or the duodenum, chemical irritants, drinking too much alcohol. When we have a bacterial infection, it's a way to clear the upper digestive tract of those bacteria. And even certain pain and psychological and sensory stimuli can induce vomiting. It usually is preceded by nausea and retching. Retching is kind of the beginning of vomiting, but it doesn't quite come up. You can feel certain things happening, the dilation of the esophagus. You can feel some chyme entering the lower part of the esophagus, but it doesn't quite come out. And then also usually you start really salivating and sweating and even some tachycardia. When vomiting occurs, those strong abdominal contractions and rising thoracic pressure force the upper esophageal sphincter or cardiac sphincter open. The esophagus and the body of the stomach relax, and chyme is driven out by pretty forceful reverse peristaltic contractions. Projectile vomiting is sudden vomiting with no prior nausea or retching, common in feeding infants. Chronic vomiting can be very destructive to the body. It can mess with your electrolytes and your acid-base imbalances. It can wear away the enamel of your teeth because of the hydrochloric acid in the vomit. Bulimia is an example. Chronic vomiting. You also have the danger of the aspiration of the vomited contents into your respiratory tract. Sometimes surgical anesthesia can induce nausea and vomiting. So usually before surgery, they'll make you fast so that we're sure the stomach and the small intestine are empty. Salivary and gastric enzymes partially digest protein and lesser amounts of starch and fat in the stomach. So most of the digestion is going to occur in the small intestine. The stomach doesn't actually really have the job of absorbing nutrients to the blood, but a few things will absorb, like aspirin and some lipid-soluble drugs. Alcohol is absorbed mainly by the small intestine, and intoxicating effects depend partly on how rapidly the stomach is emptied. The stomach is protected in three ways. Remember, it's a harsh acidic environment with other degrading enzymes. So there's the mucus coat, the tight junctions that hold the cells tightly together, and the epithelial cell replacement every 3 to 6 days. So a lot of mitosis going on, creating new cells to line the stomach. If you have a breakdown of any of these measures, you could have inflammation or a peptic ulcer. And those look like this. Some of these ulcers can even be caused by the H. pylori bacteria. Nervous and endocrine systems collaborate for the regulation of gastric function. They increase gastric secretion and motility when food is eaten. When we eat food, that's when we need those things to increase. And then we'll suppress them when the stomach empties. There's a lot of autonomic nervous system control with gastric activity. The cephalic phase is when the stomach is being controlled by the brain and includes the gastric secretions. There's the gastric phase where the stomach controls itself. Still gastric secretions and motility occur. Then there's the intestinal phase where the stomach is being controlled by the small intestine, and still gastric secretions and motility occur. And these phases can overlap or occur at the same time, and are shown right here. Ingested food stimulates gastric activity two ways: by the stretching of the stomach and by increasing the pH of the contents. The gastric secretion is stimulated by three chemicals: acetylcholine, secreted by the parasympathetic nerve fibers of both reflexes; histamine, which is a paracrine secretion from the enteroendocrine cells of the gastric glands; and gastrin, which is a hormone produced by the enteroendocrine G cells in the pyloric glands. There's those G cells right here. In this section, we'll look at some of the accessory organs. Our expected learning outcomes are to describe the gross and microscopic anatomy of the liver, gallbladder, bile duct system, and the pancreas, to describe the digestive secretions and functions of the liver, gallbladder, and pancreas, to explain how hormones regulate secretion by the liver and the pancreas. The small intestine receives chyme from the stomach and secretions from the liver and the pancreas. It can't digest everything on its own. It needs help from these accessory organs. The liver, it's the body's largest gland, weighs about 3 pounds. It's just inferior to the diaphragm and has many, many functions, but when we're talking digestive system, one of its main functions for that is to secrete bile. And bile is an emulsifier of fats. It kind of acts like soap and spreads the fat out, increasing the surface area, which again allows enzymes to get in there and do their job by breaking the fat down. The liver has 4 lobes: the right lobe, the left lobe, the quadrate lobe, and the caudate lobe. It also has the falciform ligament, which separates the left and the right lobes, and a sheet of mesentery that suspends it from the diaphragm. There's also the round ligament, which is the fibrous remnant of the umbilical vein from when the umbilical cord attached to the fetus. The blood supply of the liver is by the hepatic artery or the hepatic portal vein. When you think liver, look for this prefix on the word. The blood leaves the liver at the superior surface and immediately drains to the inferior vena cava. Then to store bile is the a structure called the gallbladder, which adheres to the depression on the inferior surface of the liver, just right between the right and the quadrate lobes. Here's the right lobe here, the left lobe. It's such a big organ that we got to look from the bottom, from the inferior view, to actually see the caudate lobe and the quadrate lobe here, and the gallbladder kind of snuggled in here on the inferior surface. Here's that round ligament, which is what attaches the baby to the placenta. Into the umbilical cord. And the falciform ligament there. We look at the microscopic anatomy here. Looks like a lot of branches. We'll be talking about the hepatocyte cells here as well, and the bile canaliculi. Also the bile ductule here. Here's the branch of the hepatic portal vein and the branch of the hepatic portal artery here as well. The hepatocytes, those are these cells right here. After a meal, they absorb from the blood anything they can here: glucose, amino acids, iron, vitamins, and other nutrients. Between meals, they're in charge of breaking down stored glycogen. And glycogen is a big carbohydrate, basically a chain of glucose molecules. So just breaking the bond in between all the glucoses then gets us glucose so that we can do cell respiration to make ATP. They also remove and degrade hormones, toxins, bile pigments, and drugs, and secrete certain things into the blood like albumin, lipoproteins, clotting factors, angiotensinogen, and other products. The bile canaliculi are narrow channels into which the liver secretes its bile, ultimately to the right and left hepatic ducts, then to the common hepatic duct, which is formed from the convergence of the right and left hepatic ducts on the inferior side of the liver, then to the cystic duct, which comes from the gallbladder and then joins the common hepatic duct, and to the bile duct, which is formed from the union of the cystic and the common hepatic ducts, and then descends through the lesser omentum toward the duodenum, and then stored in the gallbladder is the storage structure. It's kind of pear-shaped and about 10 centimeters long. It serves to store and concentrate bile by absorbing water and electrolytes. Here we can see the hepatic ducts and the common hepatic duct and the cystic duct and the gallbladder here. The head, the body, and the neck. Then we can see where that is going to, through the bile duct, empty down into the small intestine. Bile is a yellow-green fluid containing minerals, cholesterol, neutral fats, phospholipids, bile pigments, and bile acids. It also is where we send our bilirubin, which is broken down hemoglobin. Bacteria then will change that into urobilirubin, which is brown, which causes the brown color of our feces. It also stores bile acids or bile salts, which are steroids synthesized from cholesterol. Bile acids and lecithin, a phospholipid, aid in the fat digestion and absorption. If the area becomes very excessively concentrated with waste, bile could actually form gallstones. Bile gets to the gallbladder by first filling the bile duct and then overflowing into the gallbladder. The liver secretes about 500 to 1,000 milliliters of bile daily. 80% of the bile acids are reabsorbed in the ileum and then returned to the liver. Then the hepatocytes absorb and resecrete them. With the enterohepatic circulation, the route of secretion, reabsorption, and resecretion of bile acids make it about 2 or more times during digestion of an average meal. 20% of the bile acids are excreted in feces, and it's the body's only way of eliminating the excess cholesterol that could build up. And the liver synthesizes new bile acids from cholesterol to replace those lost in the feces. Gallstones are hard masses in either of the gallbladder or bile duct. It's composed of cholesterol, calcium carbonate, and bilirubin. Cholelithiasis is the formation of gallstones, very common in obese women over 40 because of excess cholesterol. They can block the removal of bile from the gallbladder, so there can be obstruction of those ducts, which then results in jaundice and yellowing of the skin, poor fat digestion, and impaired absorption of fat-soluble vitamins. Lithotripsy is the use of the ultrasonic vibrations to pulverize stones without surgery. And then they're just passed. The pancreas is another important accessory organ. It's a spongy retroperitoneal gland posterior to the greater curvature of the stomach. It's about 12 to 15 centimeters long and 2.5 centimeters thick. It has a head, a body, and a tail. As we remember from a few chapters ago, the pancreas has endocrine function in that it has islets that secrete insulin and glucagon. Those are mostly in the tail of the gland, but it also, mainly 99%, has exocrine portions that secrete 1,200 to 1,500 milliliters of pancreatic juice every day. They also have secretory acini that release their secretion into smaller ducts that converge into the main pancreatic duct. The pancreatic duct runs lengthwise through the middle of the gland and then joins the bile duct at the hepatopancreatic ampulla. Then the hepatopancreatic sphincter controls the release of both bile and pancreatic juice into the duodenum. Hopefully we'll have a picture of that coming up soon. The accessory pancreatic duct is a smaller duct that branches from the main pancreatic duct and opens independently into the duodenum. It bypasses the sphincter, and it allows the pancreatic juice to be dripped into the duodenum without the bile. Pancreatic juice is an alkaline mixture. Has the sodium bicarbonate, other zymogens in it. This is to help neutralize the hydrochloric acid coming out of the stomach. The pancreatic zymogens are trypsinogen, so that's the precursor molecule. It's secreted in the intestinal lumen and then converted to trypsin, which that is the actual enzyme that's going to break down molecules in the small intestine. The enzyme that activates it is called enterokinase. Trypsin is autocatalytic because then once we create some, it can help convert more trypsinogen into still more trypsin. Chymotrypsinogen is another one of our important enzymes, and it's converted to trypsinogen by trypsin. And procarboxypeptidase is converted to carboxypeptidase by trypsin as well. Then there's some other pancreatic enzymes. These listed up here, these are mostly in charge of breaking down, chemically breaking down proteins. These other ones, pancreatic amylase, is in charge of still the breakdown of more starches or carbohydrates. Pancreatic lipase is digesting of fat. And ribonuclease and deoxyribonuclease help to break down RNA and DNA. Remember, often when you see an ASE ending, it's an enzyme. There's those acinar cells making those precursors or zymogens. Here it shows the chart of the trypsinogen being converted to trypsin by enterokinase, and then also acting to activate these other two enzymes. 3 stimuli chiefly responsible for the release of pancreatic juice and bile. They're acetylcholine, cholecystokinin, and secretin. Acetylcholine comes from the vagus enteric nerves. It stimulates those acini cells to secrete enzymes during the cephalic phase of gastric control. Cholecystokinin, also known as CCK, is secreted by the mucosa of the duodenum in response to the arrival of fats. Secretin is released from the duodenum in response to the acidic chyme arriving from the stomach. This one stimulates the ducts of both the liver and the pancreas to secrete more sodium bicarbonate to help raise that pH level. In this section, we're going to talk more specifically about the small intestine. Our learning objectives are to describe the gross and microscopic anatomy of the small intestine, to state how the mucosa of the small intestine differs from that of the stomach, and explain the functional significance of the differences, to define contact digestion and describe where it occurs, and to describe the types of movement that occur in the small intestine. Nearly all chemical digestion and nutrient absorption, or the most significant part of it, occurs in the small intestine. It's the longest part of the digestive tract, about 5 meters long. With no muscle tone in a cadaver, it can be up to 8 meters long. And when we say small intestine, we're referring to a smaller diameter than the other one, which is the large intestine. The small intestine is a coiled mass, and it does fill most of the abdominal cavity. It's inferior to the stomach and the liver. Located right here. This part here is the large intestine. And you can see there's a lot more coils in the small intestine. The small intestine is divided into different segments: the duodenum, the jejunum, and the ileum. Here we can see the duodenum begins here. The jejunum is in purple, and the ileum is kind of in pink here. The duodenum is about 10 inches long and begins at the pyloric valve, the pyloric sphincter there. The major and minor duodenal papilla are distal to the pyloric valve and receives the major and minor pancreatic ducts. Now I've backed up to this picture here so we can see. Here's the major duodenal papilla sticking out from these ducts. It arches around the head of the pancreas and then ends at a sharp bend called the duodenojejunal flexure. Most of it's retroperitoneal, and it receives stomach contents, pancreatic juice, and bile. And our stomach acid is neutralized here. And fats are physically broken up and emulsified by bile acids. Pepsin, which worked really well in the low pH of the stomach, is now inactivated because it doesn't work as well in the alkaline or basic environment of the duodenum, and pancreatic enzymes that are dumped here into the duodenum start to perform their chemical digestion. The jejunum is the first 40% of the small intestine beyond the duodenum. It's about up to 1.7 meters in a living person. Many circular folds, it's thick and muscular, has a good rich blood supply. And the most of the digestion and nutrient absorption will occur here. The ileum is the last 60%. It's about 2.7 meters long. It's a little bit thinner and less muscular, a little paler in color. It includes Peyer's patches, which have prominent lymphatic nodules. Ileocecal junction, which is the end of the small intestine, where the ileum joins the cecum, and the ileocecal valve, which is a sphincter that's formed by the thickened muscularis of the ileum. It protrudes into the cecum. This regulates the passage of food residue now, because now it won't be digested anymore into the large intestine. And both the jejunum and the ileum are intraperitoneal and covered with serosa. The small intestine tissues are designed for nutrient absorption and digestion. Their lumen is lined with simple columnar epithelium, and their muscularis externa is noted for a thick inner circular layer and a thinner outer longitudinal layer. This is for those peristaltic contractions. Besides being a long organ, we can increase the surface area even more by having circular folds inside. It increases the surface area by 2 to 3 times. Then there's villi, which increase it by a factor of 10, and microvilli, which increases the surface area by a factor of 20. The circular folds are the largest folds of the intestinal wall, up to 10 millimeters high. They involve only the mucosa and the submucosa and occur from the duodenum to the middle of the ileum. They cause chyme to flow in a spiral path, which then causes more contact with the mucosa, and it promotes more thorough mixing and nutrient absorption. The villi are the finger-like projections, only about up to a millimeter tall, and they make things look fuzzy on the inside of the small intestine. And they're covered with absorptive or goblet cells. Goblet cells secrete mucus. And they have tight junctions so that enzymes don't seep out. And the core of the villus is filled with areolar tissue of lamina propria, which then contains arteriole capillaries, venule, and lymphatic capillaries called the lacteal. And we learned a few chapters ago that the lacteal is going to be where lipids are sent to the lymphatic system and then sent to the blood. Here's what a villi looks like. And on the inside, here's what the lacteal looks like. The microvilli form what's called a fuzzy brush border on the apical surface of each absorptive cell. They're only about a micrometer high, but it does help increase the absorptive surface area. They also make brush border enzymes. They carry out the final stages of enzymatic digestion. They're not released to the lumens, so these items need to come in contact with the brush border in order to take advantage of these enzymes. It's called contact digestion. And you have to look really close here on this villi. Right there, that little hairy surface, that's the brush border. There's duodenal glands in the submucosa of the duodenum, which secrete an abundance of bicarbonate-rich mucus. Of course, this is important for that area because the acidic stomach contents are coming and we need to neutralize it. And there's also a lot of lymphocytes there to help keep at bay any pathogens that might have made it all the way through so far. In the intestine, there's intestinal crypts, which secrete about 1 to 2 liters of intestinal juice a day in response to acid, hypertonic chyme, and the distension of the intestines. The pH is up to about 7.8. Contains water, mucus, and a little enzyme. Intestinal motility. So contractions of the intestine serve 3 functions. We want to mix chyme with the intestinal juices, like bile and pancreatic juice, to neutralize acid and to be able to digest nutrients more effectively. And we want to churn the chyme and bring it into contact with the mucosa for contact digestion and nutrient absorption, and to remove residue toward that large intestine. Segmentation is the name of the movement, in which the stationary ring-like constrictions appear in several places along the intestine, shown in this picture below. They'll appear for a while in one place and then relax, and then a little further up, then that'll constrict, and that'll help swish the contents kind of back and forth. So again, segmentation is more for the back and forth movement of intestinal contents. There's pacemaker cells in the muscularis externa to set this rhythm. Again, pacemaker cells are probably going to have a little leaking of sodium, a lot like the heart pacemaker. The contractions are about 12 times a minute for the duodenum and 8 to 9 times per minute in the ileum. When most nutrients are absorbed and little remains except the residue, the segmentation declines and peristalsis begins. And peristalsis is a wave-like contraction that pushes food forward instead of back and forth like segmentation. A peristaltic wave can begin in the duodenum, travel for 10 to 70 centimeters, and then die out. And then it'll be followed by another wave. The ileocecal valve is usually closed, but food in the stomach can trigger the gastroileo reflex that enhances segmentation in the ileum and then relaxes the valve. As the cecum fills with residue, the pressure pinches the valve shut and prevents the contents from going back. In this section, we're going to take a look at chemical digestion and absorption. Our objectives are to be able to describe how the major class of nutrients are chemically digested, name the enzymes involved, and discuss the functional differences among these enzymes. So some of this may be a little review from what we've already talked about. We should also be able to describe the type of nutrient that's absorbed by the small intestine, to describe how each type of nutrient is absorbed by the small intestine. There we go. Starch, remember that's a big carbohydrate. It's the most digestible dietary carbohydrate. Cellulose, that's in plant material. That one's indigestible. So anything indigestible like cellulose, we call that fiber, like in lettuce and such. So again, cellulose is in plant material. Starch, though, which is how plants store their glucose, the cellulose is more of their cell walls, but starch is their glucose stores in their central vacuoles in plant cells. So when we eat plants, we eat them for their starch, not for their cellulose. So starch is basically chains of glucose. They're chained in a way that we are able to digest them, unlike cellulose. So we first digest them into smaller chains called oligosaccharides, which are only about 8 glucoses long. Then we digest those down to disaccharides, which di means 2 glucoses long. We also can call that maltose. you've ever heard of malt sugar in beers and such, that's maltose. It's a disaccharide, two glucoses bonded together. And then we can break that final bond between those two glucoses and have our individual glucoses. And when we have individual glucose, we're able to absorb that into the small intestine. This process begins in the mouth with salivary amylase. It works best at about the pH of our oral cavity. Once amylase hits our stomach, when it's in an acidic environment, it's denatured. When reaching the small intestine, the pancreatic amylase takes over and quickly converts the starch to oligosaccharides and then to maltose. And the brush border enzymes finish it by breaking that final bond on the disaccharide to make glucose into its two monosaccharides. And then we can immediately absorb it. The plasma membrane of absorptive cells has transport proteins that absorb monosaccharides as soon as the brush border enzymes release them. In fact, 80% of absorbed sugar is glucose, and it's taken up by the sodium glucose transport proteins, SGLT proteins. Then glucose is transported out of the base of the absorptive cell into the extracellular fluid by facilitated diffusion. Sugar entering the extracellular fluid will increase the osmolarity, increase the pressure out there, which will draw water osmotically from the lumen of the intestine through the leaky tight junctions between the epithelial cells. And then water will carry more glucose and other nutrients with it by what's called solvent drag. This also absorbs galactose. Fructose is absorbed by facilitated diffusion. Fructose is very similar to glucose. In fact, it has the same formula, C6H12O6. Glucose, galactose, and any remaining fructose, that's the sweetest sugar, they're transported out of the base of the cell by facilitated diffusion. Facilitated diffusion is nice. It doesn't take any extra ATP to do that. It's just basically a carrier protein allowing the glucose through. It's absorbed by the blood capillaries in the villus, and then the hepatic portal system will deliver it to the liver. Interestingly enough, lactose is another sugar. It's a disaccharide. In people who don't have the enzyme to digest lactose, usually everybody has it as a baby, but many people of the world stop making lactase when they get to a certain age. So then that lactose, which again is a disaccharide, passes on to the large intestine undigested. It increases the osmolarity of the intestinal contents, which causes water retention in the colon, and then that causes diarrhea. And the bacteria in there love that lactose and will start fermenting it, which causes a lot of gas. So people who have lactose intolerance are very gassy with their large intestine and have diarrhea when they consume lactose sugar, which sometimes it's nicknamed milk sugar. It's in milk and ice cream. It occurs in only about 15% of American whites, 90% of American blacks, 70% of Mediterraneans, and nearly all Asians. So basically, this is a great example of evolution. Evolution is just changing in genes. So quite a few people in America have the mutated gene which keeps our lactase enzyme on, so we can enjoy lactose sugar all of our lives. Whereas in, for instance, all of Asians, pretty much all of them, their lactase making enzyme, that gene gets shut off, and then they can't enjoy wonderful milk and ice cream. Luckily enough, many can consume yogurt and cheese since bacteria in those products have mostly broken down the lactose. Amino acids are absorbed by the small intestine, and they come from three sources: from our dietary proteins that we eat, from our digestive enzymes, digested by each other, and by sloughed epithelial cells digested by enzymes. Wow, that's like recycling. I like it. So proteases or peptidases are enzymes that digest proteins. Again, we got that ASE ending there. They begin their work in the stomach at an optimal low pH. Pepsin digests about 10 to 15% in the stomach. Protein then gets digested further in the small intestine, where pepsin is actually inactivated, but other enzymes like trypsin and chymotrypsin will take over the process, and they'll take those proteins, make them into smaller segments called polypeptides, and even shorter ones yet called oligopeptides, until we finally get them down into their individual amino acids by those brush border enzymes, finishing the task again. Then sodium-dependent amino acid cotransporters will move those amino acids into the epithelial cells, and then facilitated diffusion will move them into the bloodstream. Infants will absorb proteins by pinocytosis, and then will release them to the blood by exocytosis. Hydrophobicity of lipids, meaning their fear of water or their inability to mix with water, makes their digestion and absorption a little more complicated, since they don't like water and we are mostly made out of water. So lipases, again, ASE ending, is a fat digesting enzyme. Lingual lipase is secreted by the intrinsic salivary glands of the tongue, active in the mouth, but more active in the stomach, along with gastric lipase. 10 to 15% of lipids are digested there before reaching the duodenum. Before digestion in the duodenum, got a little spelling error there. Vigorous pumping in the stomach's antrum will emulsify the fat and break up the globs. And emulsification droplets are then passed to the small intestine. Those will be broken further by bile and lecithin and agitation by the intestinal segmentation, which exposes more surface area to enzymatic action. It said there's enough pancreatic lipase in the small intestine after a meal to digest the average daily fat intake in as little as 1 to 2 minutes. Lipase will act on triglycerides and will turn them into 2 free fatty acids and a monoglyceride. Here's what an emulsification droplet looks like. The absorption of free fatty acids, monoglycerides, and other lipids depend on minute droplets in the bile called micelles. Those are made in the liver, and they pass into the bile duct into the duodenum, and there they absorb the fat-soluble vitamins, cholesterol, free fatty acids, and monoglycerides. Then they transport the lipids to the surface of the intestinal absorptive cells. Then the lipids leave the micelles and diffuse through the plasma membrane into the cells. The micelles can be reused, which is nice. Within the intestinal cell, the free fatty acids and monoglycerides are then transported to the smooth ER. Then they're resynthesized into triglycerides. A triglyceride is a lipid with 3 fatty acid chains. Then the Golgi complex will coat these with phospholipids and proteins. kind of make a package or a vesicle out of them called the chylomicron. Those will be packaged further into secretory vesicles that will migrate to the basal cell surface. Then those vesicles will release their contents into the core of the villus, where it can finally be taken up by the lacteal, put into the lymph, and it'll end up then finally in the bloodstream when the lymph dumps it back to the bloodstream. This chart sums up most of what we just went over and will be a good study tool for you. Nucleic acids, those are DNA and RNA. Deoxyribonuclease and ribonuclease help to digest them down into their nucleotides, which is their monomer or small form. Nucleosidases and phosphatases of the brush border will split them down even further into their phosphate items, ribose, deoxyribose sugars, and nitrogenous bases, and the membrane carriers will allow absorption. Vitamins will be absorbed unchanged. Fat-soluble ones like A, D, E, and K will be absorbed with other lipids. If ingested without fat-containing food, then they're not absorbed at all, but will be passed in the feces and wasted. Water-soluble ones like B and C will be absorbed by simple diffusion, and B12 if bound to an intrinsic factor from the stomach. Minerals and electrolytes will be absorbed all along the small intestine. Sodium is co-transported with sugars and amino acids. Chloride is exchanged for bicarbonate, reversing chloride. Bicarbonate exchange that occurs in the stomach. Potassium is absorbed by simple diffusion. Iron and calcium is absorbed as needed. Iron absorption is stimulated by a liver hormone called hepcidin, where absorptive cells bind the Fe2 and internalize them by active transport. They're unable to absorb the Fe3s, but stomach acid will reduce them to Fe2s. Transferrin, it's an extracellular protein, will transport iron in the blood to bone marrow, muscle, and liver. Calcium is absorbed throughout the intestine by different mechanisms. Transcellular absorption in the duodenum, it's where it enters through the calcium channels in the apical cell membrane and binds to calbindin, which is a protein. And so the concentration gradient will continue to favor the calcium influx. And then they're actively transported out of the base of the cell into the bloodstream by calcium ATPase and the sodium-calcium antiport. Then diffusion between the epithelials in the jejunum and the ileum occurs. And then most calcium is absorbed from meat and dairy. And dietary fat retards calcium absorption. Parathyroid hormone is secreted in response to a drop in blood calcium levels, like we learned a few chapters ago. So that'll stimulate the kidney to make vitamin D. And then vitamin D will affect the absorptive cells of the duodenum in 3 ways. It increases calcium channels and the amount of calbindin, and then the number of calcium ATPase pumps. So this will help increase calcium in the blood. Water, the digestive system is one of several systems involved in water balance. It receives about 9 liters of water a day. And in the end, about 0.2 liters is voided by daily fecal output. But the rest will all stay in the body or be removed by other methods like sweating and urination. diarrhea is when excess water builds up in the large intestine. It makes feces pass much more quickly. Constipation will occur when there's not enough water in the intestine. So we say too much water gets reabsorbed or put back into the blood, not leaving enough to keep the feces moist enough, so then the feces becomes hardened. In this last section, we'll talk about the large intestine. Our expected learning outcomes are to describe the gross anatomy of the large intestine, to contrast the mucosa of the colon with that of the small intestine, to state the physiological significance of intestinal bacteria, to discuss the types of contractions that occur here in the colon, and explain the neurological control of defecation. The large intestine receives up to about 500 milliliters of indigestible residue per day. It reduces it to about 150 milliliters of feces by absorbing water and salts back to the blood, and then it eliminates that feces by what's called defecation. The large intestine is about 5 feet long, loops up and around in our abdomen, has a bigger diameter than the smaller intestine. It begins as what's called the cecum, inferior to the ileocecal valve. The appendix is attached to the lower end of the cecum. It has a bunch of lymphocytes in it, which is a significant source of immune cells, but if it becomes inflamed and infected, you can have it removed and still be fine. There's the ascending colon, the right colic or hepatic flexure, the transverse colon, the left colic or splenic flexure, and then the descending colon. And this all kind of loops up and around and frames the small intestine. The sigmoid colon is S-shaped. It's a S-shaped portion leading down into the pelvic cavity. The rectum is the portion ending at the anal canal and has 3 curvatures or 3 infoldings. Then there's the anal canal. It's the final 3 centimeters of the large intestine. It passes through the levator muscle and the pelvic floor and terminates at the anus. There's the anal columns and the sinuses, which exude mucus and lubricant into the anal canal during defecation. There's large hemorrhoidal veins or superficial plexus in the anal columns and around the orifice. And the hemorrhoids are permanently distended veins, can be very painful, that protrude into the anal canal or bulge outside of the anus, especially when they're inflamed. The muscularis externa of the colon is unusual. It has the Taenia coli, which are longitudinal fibers concentrated in 3 thickened ribbon-like strips. The haustra, which are pouches in the colon caused by the muscular tone of the Taenia coli. There's the internal anal sphincter, which is smooth muscle, and the external anal sphincter, which is skeletal muscle. And you already know that with skeletal muscle, then you have voluntary control. So at least that one you can control. There's omental appendages, which are like fatty pouches, but they're not really known why they're there. So we can locate all of those structures here, beginning at the ileocecal valve here, going up from the cecum and the appendix here, the ascending colon. There's the haustrum, the transverse colon with the right colic flexure there, the left colic flexure here. This is the descending colon, where we can see the Tenei coli. Remember the mesocolon we talked about earlier, and the omental appendages. Down here, this last little loop is the sigmoid colon. We reach the rectum, and then the anal canal, and the locations of the sphincters. Here we get a closer look of the rectum, the rectal valve here, the anal canal. Here's the hemorrhoidal veins, the sphincter, internal sphincter, and the external sphincter. There's a lot of bacteria in the large intestine, about 800 species of bacteria. They help to digest cellulose and other big carbohydrates, which actually helps to synthesize vitamins for us, like B and K vitamins. But they also, through their digestion themselves, they make an intestinal gas called flatus. You might make about 500 milliliters per day. Most of it is reabsorbed. Some of it can be swallowed air, but the bacteria add to it. Can contain hydrogen sulfide, indole, and scatol, which are what produce the odor. This is interesting. During surgery, surgeons need to watch their flames. If any electrical cauterization is going on, that hydrogen gas in the intestine could actually explode. The large intestine takes about 36 to 48 hours to reduce the residue of a meal to feces. Most of the time is spent in the transverse colon, but really there's no chemical change in the residue. We're working on reabsorbing water and electrolytes back to the blood. Feces does consist, though, about 75% water and 25% solid. The haustral contractions occur every 30 minutes. Distension of a haustrum stimulates it to contract, churns and mixes the residue, promoting water and salt absorption. And the mass movements are stronger contractions that occur 1 to 3 times a day. If you're lucky, right? It's triggered by the gastrocolic and duodenocolic reflexes. Filling of the stomach and the duodenum can actually stimulate motility of the colon. That way you move out the old and make room for the new. Stretching of the rectum stimulates 2 defecation reflexes. It accounts for the urge to defecate, often felt soon after a meal. The intrinsic defecation reflex works entirely within the myenteric plexus to produce relatively weak response. The stretch signals travel through the plexus to the muscularis, causing it to contract and the internal sphincter to relax. So look at the internal sphincter you do not control. That one is controlled for you. Parasympathetic defecation reflex involves the spinal cord. The stretching of the rectum sends sensory signals to the spinal cord, and pelvic nerve returns signals, intensifying the peristalsis and relaxing the internal anal sphincter. But it occurs, defecation, only if the external anal sphincter and puborectalis muscles are voluntarily relaxed, meaning you control that. Using the Valsalva maneuver can help remove the feces. And here we see a diagram of that with the neural control included here. The sensory fibers and the voluntary motor fibers innervating the external anal sphincter. I guess you could say you should be thankful for your sphincters. That way you have the ability to control when and where this happens. Infants don't have that control right away, but must develop it. Your book has a neat story about the man with a hole in his stomach. I actually remember reading this story when I was in elementary. A Canadian was shot accidentally by a shotgun in 1822 with a hole large enough to put one finger in, and as it healed, it was covered with a flap of tissue that you could flip up. And so this physician, William Beaumont, experimented on him. He would put samples of food in there, tied to string, and would remove the samples, make observations, and this would help figure out how long it takes to digest items. He published his book in 1833, and it laid the foundation for modern gastric physiology. Unfortunately, here's an example of where knowledge came to be through someone's accident.